What up, YouTube? This is Joe Colletti and Kevin Curran coming at you with a brand new video about CRISPR. Amazing technology. Can't wait to get to you. So let's get it started.
All right, Joe. So tell us, what is CRISPR?
So CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats.
I mean, that's a mouthful, but what does it actually mean?
Basically, CRISPR is a gene editing system that is found in different types of bacteria to fight off viruses. It works with an enzyme called Cas9, which stands for CRISPR-associated endonuclease, that deletes parts of the DNA strand.
I mean, that's a lot of information right there. Hope you guys enjoyed that part. All right. According to the U.S. National Library of Medicine, CRISPR was first found in bacteria fighting viruses. It does so by capturing pieces of the virus's DNA and cloning these segments to create a new strand called a CRISPR array. This allows the bacteria to recognize the virus if it ever decides to return.
When and if it does return, this new array is used to recognize a virus and then go in and create a matching RNA strand, which then binds to the Cas9 enzyme and goes in and finds the virus and breaks down its DNA, disabling it.
So, Joe, where did we find all this?
Well, Kevin, according to the Broad Institute, in 1993, a Spanish scientist and microbiologist named Francisco Mojica discovered this process in his research in the Halloveric species of bacteria. The idea of an adaptive immune system gained attention around the world for the scientists.
Around the same time, in 2005, a French scientist named Alexander Bolton discovered the Cas9 enzyme. These discoveries allowed us to fully understand the CRISPR and gave us the potential to use it in the modern world.
So, what is it now?
After years of development, CRISPR is a gene editing tool that professionals can use to alter DNA by modifying, deleting, or inserting information into the genome.
This picture represents what CRISPR means to us now very well. Like a surgeon, the system can hack into the DNA strand and specifically clip out nucleotides in order to alter the genome.
I mean, that is pretty cool and all, but how does this actually work?
Glad you asked, Kevin. Let's get into the details of how this mechanism actually works and functions. CRISPR is a two-part system. The first, most important and critical part of the system is a Cas9 enzyme. The enzyme is able to work directly with the genome by binding itself to the DNA. Cas9 can then break the polypeptides apart, causing a double helix split. This is what allows CRISPR to edit specific sections of the DNA.
The second part of the system is the guide RNA. The guide RNA is a piece of RNA complementary to the DNA being affected. The strand directly allows the Cas9 to edit and change strands of the DNA.
The Cas9 enzyme binds together with the strand of guide RNA to form what is called a complex. The complex has the ability to dive into the genome and locate specific targeted segments of the DNA. They locate the specific part and bind to the DNA strand, preparing to begin the editing process.
This complex then binds to the DNA strand and allows the RNA to connect to that same strand. After this is completed, the guide RNA is able to connect with the target sequence and remove it entirely from the strand. It is also able to interchange parts of the strand or target specific nucleotides.
CRISPR can edit the genome in three main ways: disruption, deletion, and correction. The first and most simple process is disruption. When the Cas9 enzyme attaches to the DNA strand, the guide RNA attaches itself to a few nucleotides, taking them out of the genome. The Cas9 complex then can unbind from the double helix, and the two loose ends come together and bind, completing the process.
The second of these three processes is deletion. Deletion uses the two guide RNA strands and repeats the process at two separate points in the genome. The two complexes of deletion allow the process to delete a much larger piece of the DNA. After this is completed, just like disruption, the two ends meet and bind to restore the strand. This allows for the entire middle piece of DNA to be disregarded.
The final process is correction, and is the most complex of all the processes. It involves only one Cas9 complex, but it does more than breaking the nucleotides off the genome. This process is used to fix mutations by replacing incorrect nucleotides. The guide RNA will code for the correct nucleotide and insert them into the genome. This image is a good overview of how the CRISPR process works. As you can see, the RNA is inside the Cas9 enzyme and wraps itself around the DNA strand. The RNA attaches itself to the DNA strand and codes for the new modified piece of DNA. So, we talked a lot about CRISPR, where it came from, and how it works. But why is this so important to us?
Well, now we really want to discuss how we can use this in everyday life. Scientists have been discussing about using CRISPR in the agricultural fields, the biological fields, and even the medical fields. There are endless opportunities to use this technology.
There are two main ways that scientists have been discussing using the CRISPR technology. The first and obvious way is using it in order to modify errors in the genetic code of organisms and the genetic diseases.
The second way researchers have also discovered is using modified Cas9 to access and change epigenetic information. This can be done by altering and activating or deactivating parts of the genome. Alright, so one way we are using CRISPR in the medical field is to fight various deadly diseases that have been found in multiple patients all across the country and the world. These diseases have really taken a toll, and the fact that we can use CRISPR to maybe cure them is amazing.
For example, according to the website Labiotech, Chinese doctors have been testing the CRISPR technology on patients with severe esophagus cancer. They've been extracting T cells from patients and using the technology to remove genes that code for receptors that cause tumors. Over 80 patients have been treated, with pending results.
Another disease in which scientists have identified that we could possibly use CRISPR to help cure is cystic fibrosis. So researchers have actually recently identified the exact gene that affects this respiratory disease, and they discovered a way in which we can use CRISPR in those cells. The only problem currently is we have not tested it on humans, and who knows when that will happen due to strenuous regulations. Some other diseases that CRISPR could actually be used to help cure in the future are AIDS, various blood diseases, and one day it might even be used to help cure blindness in humans.
According to the website Popular Mechanics, CRISPR can be used in a similar way to affect farm animals. Using the technology to affect the genome in order to change the growth rate of hair and muscles, Chinese scientists have already used this technology to increase their yield of wool from sheep and meat from cows.
So another way we can use CRISPR in the agricultural field is to go in and edit the genes of various different crops all across the world. With CRISPR, we have the ability to change the genome of the crops to be resistant to outside factors. This is way better than using any type of pesticide due to the fact that many pesticides have harmful effects to the environment. For example, recently researchers have used CRISPR to genetically modify grapes to resist a downy mildew, which is a harmful type of grass that often grows in the same area as grapes.
With all of CRISPR's advancements, it's hard not to think, can we use this technology to change the outcome of our children's physical appearances? According to Technology Review, this idea is very possible, and allowing parents to potentially pick their baby's eye color and hair color is definitely possible.
Cool beans! Alright, now CRISPR's cool and all, but let's talk about some controversy over the subject. You know, nothing can be perfect, and CRISPR is not one of those things.
So what's wrong with CRISPR? Many people believe that CRISPR and Cas9 will someday be the next big thing, but a lot of people are opposed to it. The introduction and use of CRISPR and Cas9 brings about many social and ethical issues about the... Sorry about the technical difficulties, but there is still much social and ethical issues with the use of CRISPR on humans.
According to this graph from the Technology Review, about half of American adults think it's okay to use CRISPR in order to reduce the risk of serious disease in children. However, at least 83% of American adults think it's not okay to use CRISPR in order to make a baby more intelligent.
So, Joe, why do we have some issues with it?
Well, according to the Journal of Clinical Research and Bioethics, for success of CRISPR and Cas9, we need the benefits to outweigh the risks. One of the major problems is the application of CRISPR and Cas9. It may cause off-target mutations in the sequence, and the mutations can be very damaging. Mutations may also cause cell death or transformation, which can be devastating to humans.
No good.
So, Dr. Barboza, why is this information so important to us?
The development of this technology could be extremely ground-changing with all the opportunity it gives. The open-endedness of CRISPR allows scientists the ability to potentially fix a plethora of issues in the medical field, as well as tons of other areas of study. CRISPR has the potential to replace harmful drugs and chemicals used on a daily basis, like pesticides, fungicides, chemotherapy, and much more.
Thank you so much for that insight, Dr. Barboza. And Dr. Barboza is not an actual doctor. Alrighty, just to wrap things up real quick, CRISPR is a recently discovered gene editing tool that is used to change the genetic code of organisms' DNA. The potential uses for this new technology in the future range from saving a life, to changing our physical appearance, to aiding in our agricultural and livestock needs. CRISPR needs a lot more research before we can begin using it, but with that said, the future with this technology is looking really bright. Alright, thank you guys for tuning into this video. I hope you guys liked it and enjoyed it and learned something from it. So please like, subscribe, and comment on our latest video. And now for a little outro music by our very own Joseph Coletti.